Thanks to all of you for coming online today for the webinar. And OutsideIn is a thematic webinar series that had been started since the summer, and ever since we have had a lockdown. And this is really meant for high school children, young adults, and also, old adults. So this is meant to really give a glimpse to the outside world about what's happening in the labs. But having said that, we chose the theme of ecology. The major theme is ecology. So I'm happy to say that starting from the summer, we've had different experts in the area give you a glimpse on different topics in ecology. Now, what we are trying to do is to imbibe these ideas a little more carefully. So last month, we had the theme on biodiversity, and this was co-hosted by Uma. And I'm very happy to say that Dr. Deepa Agashe is co-hosting this month's talks, and that means 4 talks, and it is going to be on the topic of microbes. With this, I would like to request Deepa to introduce the speaker for today. Deepa, please.
I think I was muted. Thank you, Sowdhamini. It's really fun to be part of this series, and I'm particularly excited this month because I get to welcome 4 very exciting speakers who do very, very interesting work themselves and have a broad understanding of the field to be able to tell us a bit more about different aspects of microbes and their natural habitats and what they do and how they survive and how they live. So today's speaker is Dr. Primrose Boynton. Primrose, I know her since she was a graduate student at Harvard University when she was studying the fungi, the ecology of the fungi that inhabit pitcher plants, which are these carnivorous plants that many of you have probably heard about before. And she was doing very interesting experiments with natural pitcher plant populations, which I hope she'll touch upon a little bit today, if not in detail. And then she went on to do a postdoctoral. She was a postdoctoral fellow at the Max Planck in Plön in Germany. And she's probably going to tell us something about what she's done there. She continued investigating the life of yeasts of different species and their diversity and their ecology in the wild. And now she has recently started as a faculty at Wheaton College in Massachusetts. And she will continue, I think, to do research to understand microbes and their ecology and evolution, focusing, I think, on fungi and yeast specifically. So welcome, Primrose. We're excited to have you and hear from you. So if you want to share your screen, we can get going. And as Pavitra mentioned, if those of you in the audience who are listening in, if you have burning questions, please put them in the Q&A during the talk if you want some clarification, without which it's hard to understand. And I will find a moment where it'll be appropriate to ask Primrose these questions. Otherwise, we will, of course, have plenty of time in the end for questions and discussion. So over to you, Primrose.
Thank you so much for that very friendly introduction. And I just want to tell you how honored I am to have been invited to come and talk to you. And I really appreciate all of your interest in work on microbes and in the stuff I'm about to tell you about yeast. So I'm going to be talking about yeast, yeast diversity, and I'm really interested in yeasts in both wild and domesticated habitats. And I'm going to start out by telling you a story about yeast in a domesticated habitat. So if you think about yeast, you might think about a situation like this. Yeast are responsible for making fermented beverages, things like beer and wine, and actually a lot of different alcoholic beverages. And these are very important culturally. They're very important economically. They're a part of life for many people. And it's really a yeast that's responsible for taking something from the plant world, something made of sugar, and converting it into alcohol and an alcoholic beverage. And we're going to talk a little bit through how the yeasts in our alcoholic beverages relate to the yeasts in nature. And first, I want to ask, what is a yeast exactly? And the answer is very simple. A yeast is a single-celled fungus. But if you're like me, you might find it hard to think about a fungus, which, you know, maybe you're thinking about a mushroom like the one in the slide. It's kind of a big, you know, fleshy thing. And to project your mind into what it means to be a single-celled fungus. And it might help to think about what is a fungus. So fungi are actually a really large and diverse group of organisms. People call fungi a kingdom. And when they say this, they're thinking of other kingdoms like plants and animals. Fungi are like animals, so they have to eat their food. They don't do photosynthesis. They don't make food from the sun. They're very close relatives of animals. And as you can see from the photographs of, big macroscopic fungi, they can be very diverse. And they can live in all sorts of habitats. And they really have two growth forms. If you look at a fungus up close, you're going to either see filaments, long, thin threads that are made of chains of cells growing together. And these filaments can grow together in a form like the lichen or the mushroom you're seeing. This is kind of a bunch of filaments mashed together into a shape. And these make the big fungus shapes that we're used to seeing. Or fungi can grow as single cells. And it turns out there are a lot of different single-celled fungi. There are a lot of different yeasts. And so now that you know what a yeast is, and I hope you have a vague idea at least of what a fungus is, we can ask three questions. We can ask how diverse they are, how widespread they are, and how they got into food and drink. So how diverse are yeasts? Well, I'm going to illustrate that with this picture, and this is what's called a cladogram. And this is a picture that illustrates relationships, genetic and evolutionary relationships among different organisms. And this particular cladogram is a broad representation of the kingdom of fungi. So you can see here we have a whole bunch of fungi. They all have names. Don't worry about their names. Down at the bottom, we have animals, because animals are very close relative to fungi. And the people who made this figure, they shaded in groups of yeasts. So to interpret this cladogram, you would compare how long the branches are that connect any two fungi. So these two at the top, this Lacaria and Galerina, these are both kinds of mushrooms, and they're very closely related. But to compare Lacaria and Saccharomyces cerevisiae, which is the most famous yeast, you'll see that they're actually very distantly related. And I say this because I can follow these lines down and across and back up and say, oh, that's a lot of distance. So what I'd like you to really notice in this picture is that there are at least 5 groups of yeast. And these groups of yeast are really quite different from one another. And they're often close relatives to something, in the case of these two, like mushrooms. So we have many different kinds of yeast. Yeast have evolved. The single-celled habit has evolved many times. And so we can say that yeasts are quite diverse. They really cover all of the fungi. And now I want to tell you a little bit about how widespread yeasts are. How widespread are yeasts in nature? Well, it turns out they're everywhere. If you can imagine a habitat on Earth where a yeast might grow, there is a kind of yeast that lives there. We have yeast in domesticated habitats. We have yeast that live on the bodies of animals, sometimes inside, sometimes outside. That includes human beings. There are yeasts that simply live on human beings without ever causing any symptoms. There are some other yeasts that live on or in human bodies and can make us sick. There are yeasts that live on plants. There are a lot of different yeasts that live on plants. There are yeasts that live in soil and water. And one reason you find yeast everywhere is there are so many different kinds of yeasts. So if a microbe needs a special adaptation to live somewhere, it's really likely that there's a yeast that has that adaptation. So I really want you to think about how widespread yeast are, and I want to give an example. So Deepa introduced me and talked about my PhD work on carnivorous plants. I'm not going to talk about it that much, but I did want to use it as an example of a place where you find yeasts. So here's a picture of a carnivorous plant. This plant lives in a bog and it captures insects and it uses those insects to fertilize it. And it makes these cool little leaves. The leaves fill it with rainwater and the insect can fall in. And if you take some of that water out and you spread it on a petri dish, you'll see these little colonies. And these colonies are yeast colonies, and there's different kinds. And if you were to look at the cells in one of those colonies under a microscope, you might see something like this, where you see these single-celled fungi. These little cells, you can see some of them are reproducing by making little buds. They're very cute. And it turns out it's not just carnivorous plants, it's pretty much any plant that you can sample, and you can find yeasts on it. So I hope now you have a very general overview of yeasts and why we might care about them, how diverse and widespread they are, because I'm going to be talking now about how they get into our food and drink. And I'm really going to be mostly talking about alcoholic beverages, because that's what I have the most experience with. And fermented beverages, alcoholic beverages, are really old. And what I'm showing you here is evidence from archaeology. So archaeologists go to archaeological sites and they dig up artifacts. They can only find artifacts that can survive for a long time. So that's why you usually see things like pottery. You don't see things made of wood or plant or animal materials because those just don't last that long. So of the things that people have been able to find, the oldest evidence of fermented beverages comes from China. People have found bits of pottery, and they found the chemicals on the pottery that show that the pottery was used to hold plant material and yeast and to make alcohol. And people have also found more recent artifacts of winemaking in Georgia, in Western Asia, beer making in China and Iran. And of course, nowadays, fermented beverage making is global. You know, people do it worldwide. But this is a really ancient practice. So human beings have been somehow taking advantage of yeasts for thousands of years, maybe more. So I'm going to be telling you two stories about fermented beverages. I'm going to be telling you a story about wine and one about beer. And I want us to think about what exactly happens in a fermented beverage. And the process is actually pretty simple. The details are complicated, but the idea is simple. Something has sugar in it. This something almost always comes from plants. So in the case of wine, it's grapes. In the case of beer, it's cereals like barley or wheat. A yeast converts those sugars into alcohol. And human beings kind of control that process and make a product that they want to sell. And so I'm going to tell you a story about wine. And we can simplify the process of winemaking into these three steps. People grow grapes. Grapes are grown on a farm in an agricultural environment. They harvest those grapes. They process them somehow to get the juice out. They put all of that juice into a big fermenter, and they wait. And while they're waiting, microbes in the grape juice are converting that grape juice into alcohol, and the sugars in the grape juice into alcohol to make wine. One important point about harvesting grapes is that this is a natural, this is an agricultural process. So the grapes, they're coming in contact with a lot of things from the environment. A farm has soil, right? Plants are growing in soil. So you get the grapes come in contact with the soil. They come in contact with the human workers who are picking them, with the machinery that the workers are using, with the surfaces of the grape plant themselves. Vineyards often have natural environments right next to them, things like forests. So there are a lot of places from which microbes can come. And as a result, these grapes have a microbial community already associated with them. And this microbial community includes bacteria, yeasts, archaea, other kinds of microbes like protists, all that come from the environment, from the soil, from the humans, from the machinery. It all gets into that big fermentation vessel. And some colleagues and I went to one of these fermentation vessels, and we just asked, how does the yeast community, specifically the different kinds of yeast, how does that change over time during fermentation? And we use DNA sequencing for this. So here I'm showing you a little plot on the x-axis of the plot. So here is time, is the amount of time since the workers came, mashed up the grapes and put them into the fermenter. And on the y-axis is the proportion of different kinds of yeast. So these yeasts have obscure names that you don't have to pay attention to. What I would like you to pay attention to, though, is the different colors. Just that there are a lot of different colors early on. And this represents all the different yeast coming from the environment. And in particular, they're probably coming from soil. This is probably yeast from soil that gets onto the surface of grapes. And as you can see, there's this one brown yeast, and it increases or decreases in frequency, but eventually it takes over the community. We don't really see much else besides this one brown yeast. Well, this brown yeast is called Saccharomyces cerevisiae. And this is the yeast that you most frequently find in alcoholic beverages. So here we're seeing an example of competition where this Saccharomyces cerevisiae is producing alcohol and it's producing heat because fermentation it's a hot process. And the other yeasts in the fermenter can't tolerate that much heat or that much alcohol, and they die off. And so we say that Saccharomyces cerevisiae is perhaps not present in very many numbers in the beginning, but it's a very good competitor, and it outcompetes all the other yeasts. When we looked in different fermenters, we saw this pattern over and over again. And we're not the first nor the last people to see this pattern in wine. Pretty much everyone who looks at diversity of microbes or yeast in wine sees the same thing. This is such a reliable process that people have been doing it for thousands of years. This is how wine is made. And it's reliable. A winemaker can invest a lot of money in land, plants, machinery, workers, and be very confident that every season they're going to get wine at the end. So this just happens every time. But here's an example of starting out with a lot of biodiversity and ending up with just the yeast that you want. And really kind of by luck and nature. This is how it ended up, where we could make this very economically important and culturally important product. So this is the story about wine. This is where the yeast comes from in wine. It just comes from the natural environment. All of these yeasts live out in the natural environment in the winery and in the vineyard. But the wine itself will select for the yeast that is well suited to making a lot of alcohol and tolerating a lot of alcohol. So that's a story about wine. And now I'd like to tell you a slightly longer story about beer. The general idea behind beer making is more or less the same. You get sugars from a plant material and there's yeast and it makes those sugars into alcohol. But the details are different. So people harvest cereal, and it's often barley. They'll germinate it because the embryo of the plant will convert the starch into sugar. So this is how we free up the sugar in barley. And then they cook that barley. And they cook it in kind of a barley soup. And the cooking will change the flavor, but more importantly, it'll kill off all the microbes. So we're not looking at a situation like wine where you start out with a whole bunch of microbes. Here you start out with 0 microbes because you cook that barley at a high temperature that kills off all the microbes. So then you add in some yeast. You take that cooked barley, you add some yeast, you put it into a big fermenter, and fermentation happens. The yeast converts the sugar to alcohol. There are some other processing steps, but eventually you can market your final product. Right? So the steps are the same, but the details are different, and the devil's in these details. And this really begs the question, well, okay, yeah, the yeast gets added by humans, but, you know, originally, where did that yeast come from? Did it come from nature? And to talk about this, we need to talk about the fact that there's actually 2 different kinds of beer, and they're fermented by 2 different yeasts. There's ale and there's lager. And ale is much older than lager in terms of its relationship with humans. They brew at different temperatures. That's one of the big differences. But there's also 2 different yeast species that are responsible for them. Both of these yeast species are in the same genus, so they're close relatives of one another. They have different names, and they're not the same species. So ale is brewed by Saccharomyces cerevisiae. This is the same species that we saw in wine. So it seems as though originally ale yeast probably came from the same place wine yeast did. but people have been transferring yeast, even without knowing it was yeast, just transferring stuff from beer over and over and over again for a very long time. So the story for ale might be pretty simple, and it's probably related to wine or perhaps bread making. You also see Saccharomyces cerevisiae show up in bread making, and I'm not really talking about bread making in this talk. Lager is more mysterious though. For a very long time, we did not know where Saccharomyces pastorianus came from. You don't see it in wine. You don't see it in nature. And so I want to kind of talk through the story because I think it's super interesting and fun. And it's really a human history of beer yeast. So I'm going to take you through this timeline piece by piece. And we'll start out, with something you've already learned, which is that people seem to have been making beer for thousands of years. And thousands of years ago, they almost certainly were making ales with Saccharomyces cerevisiae. And they're doing something called repitching, where you take the scum from the top of the beer, and the next time you make beer, you just put a little bit of that scum in, and it's got the yeast in it, and that can ferment your beer the next time, over and over and over again, essentially for thousands of years. This has, sustained people's beer needs for a very long time. But in the Middle Ages, in southern Germany, in a place called Bavaria, people started to make what they called lagers. Lager means cellar. It means like storeroom, because lagers are brewed at cooler temperatures. People who enjoy beer might tell you that it has a different flavor. It's less prone to contamination from microbes that'll make it go bad, mostly because it brews at a cooler temperature. And this became very popular, especially locally in Germany. But people still didn't know very much about yeast. They had a name for it, but essentially that name meant scum you get from beer. In the 1670s, microbes were discovered. And we attribute the discovery of microbes to this guy. His name is Antony van Leeuwenhoek, and he's from the Netherlands, and he built a very powerful, although very simple microscope. He peered at pond water and said, "Holy smokes, the world is full of millions of swimming tiny animals," which was just a wild idea at the time. The world is full of millions of invisible tiny animals or bacteria. And he in fact looked at yeast under the microscope, but he didn't recognize that it was a living thing. He got some from a brewer and he said, these are weird little balls you find in beer, but you probably don't want those in your beer. That's kind of weird. So, we knew about microbes, but we didn't know that yeast was a microbe. We didn't know it was a living thing until like over 100 years later. People figured out that yeast is a living thing in the 1830s, and they really did it by sitting in front of a microscope and watching yeast cells divide and make more cells. Here I'm showing you a little drawing by Louis Pasteur. He did a lot of work figuring out that ale yeast and lager yeast are really two different things. And all this happened during the Industrial Revolution. And the Industrial Revolution was a really big time for beer making because brewing was industrialized. It turns out that brewing, especially brewing of lager beers, is very well suited to industrialization. Especially if you have refrigeration. So if you can start a brewery and you have refrigeration, you can make tons and tons of lager. You can sell a lot of lager. And this really you started to see in the 19th and 20th century. Going on to the end of the 20th century, the first genome, so the DNA sequence that codes for genetic information of a eukaryotic cell. So this is a cell that has a nucleus or a cell that's not a bacterium. That first genome of a non-bacterium was the ale yeast, with Saccharomyces cerevisiae. So this is the first sequence of something like a human being, the whole genome. And this was in 1998. And that was really cool. This is a big day for yeast biologists. And all through the 20th century, people started to find and identify wild yeasts. And I'm going to tell you a little bit more about my work with that in the 21st century. And then finally, 9 years ago in 2011, the lager yeast was characterized. So here we have thousands of years of yeast history and a mystery, right? This lager yeast mystery. And I'm going to tell you first about some of my work with wild yeast, and then I'm going to tell you about how work with wild yeast, not mine, but other people's, helped us to characterize this lager yeast. So here is another cladogram. Remember, a cladogram is a diagram that illustrates how closely related different organisms are to one another. And this describes all of the Saccharomyces species that we know about today. And you can see at the top, we have Saccharomyces cerevisiae. This is ale yeast. It's also the yeast you find in wine. It's also often found in bread. And I have a red arrow pointing to a yeast called Saccharomyces paradoxus. It's the closest wild, completely wild relative of Saccharomyces cerevisiae. There are wild and domesticated cerevisiae. There are only wild Saccharomyces paradoxus. And that's the yeast that I've been working with in Germany. And we can find this yeast out in the forest. So there's a colleague of mine, her name is Doreen Landerman, and she's collecting leaf litter. So these are the leaves that have fallen off oak trees in the fall. And we can take these leaves back to the lab and we find that Saccharomyces paradoxus is growing on them. Here's another colleague of mine. She's also sampling leaf litter. Her name is Vienna Kowalik. And she found that you can find Saccharomyces paradoxus in the forest all year round at a more or less the same numbers of cells. So here we have the month that Vienna sampled. We have different trees. So she sampled next to 6 different trees. And then we have the number of cells per gram. This is on a log scale. So it goes from 10 to 100 to 1,000 to 10,000. And she was consistently finding 100 to 1,000 cells of Saccharomyces paradoxus per gram of leaf litter all year round. We thought they might die off in the winter, but they really don't. They just kind of stick around. And this led me to ask, well, is it the same Saccharomyces paradoxus? Or do you maybe see different relatives from the same family and different seasons? And so Doreen Landerman and I went out and we sampled leaf litter next to eight oak trees in Germany in different seasons. And we looked at patterns of DNA. So this is patterns of their genetic information. And we made, this is actually called a phylogram, but it has a lot in common with a cladogram. Again, you can tell how closely related any 2 individuals, and these individuals are represented by these colored bars. You can tell how closely related they are by how far you have to go down these lines to connect the 2. All of these are identical, right? You don't have to go down anywhere to connect them. But we have some more distant relatives too. About half of the ones we found were in fact all identical to one another. We found some diversity, but we didn't find that some, and we're going to call these genotypes, some genotypes were found more often in some seasons than others. So we found a lot of what we call clonality. Yeast can reproduce just by making an exact copy of itself after a cell division. So we found a lot of evidence of that. We also found a lot of evidence of inbreeding. So yeast can have sex, and it does have sex. Saccharomyces paradoxus generally has sex only with its siblings, only with its brothers and sisters. And this is just the biology of this particular species of yeast. And we found evidence of that. So we found out of 192 total individuals, we found 1 individual that was not inbred. 1 individual where we could find unrelated genetic information from 2 different parents in the same genome. So in the genetic information of that cell. So we find some evidence of sex, but this evidence points to inbreeding. We find a lot of evidence of just making a copy of yourself, asexual reproduction. And we found a lot of cool ecology that I don't have the time to tell you about, but I can answer questions about. Like some of them secrete toxins that will kill other Saccharomyces paradoxus. And some of them carry viruses that infect fungi. We found that they interact with bacteria and the trees and the other yeasts. What we can't figure out is what they eat in the forest. We expect them to eat sugar, But leaf litter is not a very high sugar environment. So there's a lot of open questions about the ecology of this yeast. So this is one Saccharomyces species. Saccharomyces is just one group of yeast in the broader diversity of single-celled fungi. But it happens to be the closest relative, Saccharomyces paradoxus, to the best characterized domesticated yeast, Saccharomyces cerevisiae, which is responsible for making ale. So let's go back to our human history of beer yeast. Here we are in the early 21st century, and we're following on the work of the late 20th century, and we're looking for Saccharomyces yeast in nature, in part because, we want to understand something about the biology of domesticated yeast, right? We're studying wild yeast to understand wild yeast, but also to help us understand domesticated yeast. And, I still haven't explained what's up with lager yeast. Where does it come from? And again, people didn't know this until the 21st century. And this is a remarkable question because lager yeast are extremely economically important. Here's a pie chart that represents the global beer value in 2014. The blue colors are lagers. The warm colors are ales and some other beers. This is over 3/4 of all global beer value. This is extremely important economically. And yet, again, in the 20th century, we simply didn't know where that yeast came from, except by saying, well, we know it's been around since Germany in the Middle Ages. One interesting thing about the lager yeast, it's called Saccharomyces pastorianus, is unlike Saccharomyces cerevisiae, it can't have sex. So remember, I told you that the yeast I study, the wild yeast Saccharomyces paradoxus, it can have sex, it does have sex, but mostly it inbreeds. It has sex with its siblings. Saccharomyces cerevisiae can have sex with its siblings, often does, also has sex with other unrelated Saccharomyces cerevisiae. Saccharomyces paradoxus does not have sex. So sex in yeast works a lot like sex in animals, like humans. The yeast will make gametes. These are like sperm and eggs, just like humans make sperm and eggs. And the gametes will fuse, they'll mate to make the new body of the organism, right? A new cell in the case of a single-celled fungus. Cerevisiae can do this. Saccharomyces pastorianus can make gametes, but the gametes are dead. We call these inviable gametes. So those gametes can never go on to find a mate, can never fuse, can never make a new body. Saccharomyces pastorianus has been reproducing asexually by cells making exact copies of themselves since at least the Middle Ages. We have very good evidence for this. And you might be familiar with a similar situation like this. And the situation you might be familiar with is the story of a mule. So a mule is, it's an animal. It's the offspring, so the child of a donkey and a horse. The problem is that donkeys and horses are not the same species. They can mate, they can make a baby mule, but the mule can never make other babies. Life for the mule stops with the mule. It can't make baby mules. It can't have sex with a donkey or a horse and make something in between. That's it. And we say that the mule is a hybrid. And we say that the mule is infertile because it can't reproduce. It can't reproduce sexually. And we say that the donkey and the horse, because they can't make a fertile offspring, that they are two different species. And this the mule resembles Saccharomyces pastorianus. It also can't reproduce sexually. So we have a good idea that it might also be a hybrid. It might be the offspring of two not terribly closely related different yeast species. So this is kind of the scenario we're thinking about, that the ale yeast and some other yeast had sex, made an offspring. This offspring was the lager yeast, and this lager yeast can't have sex again. Although again, it can reproduce by dividing its cells. It can reproduce asexually. So remember how I told you that ale yeast happens to be the first sequenced eukaryotic cell. Because we sequenced its genome, we know that ale yeast has 16 chromosomes. And here I'm just showing you a bunch of black lines that represent those chromosomes. It has 16 chromosomes. Its diploid cells have, of course, double that, 32. And so what happens if we sequence Saccharomyces pastorianus? In the last 30 years, DNA sequencing has become cheaper and cheaper and easier and easier. And nowadays, it's not a big surprise to take a microbe and sequence its entire genome. And so people did this in 2008. And here's what they found. They found that lager yeast has a lot more chromosomes than ale yeast does. And these chromosomes, if you line them up to the ale yeast, so remember the ale yeast is this black chromosome, you can line up the chromosomes of the lager yeast, and they mostly line up really well. The genetic information mostly matches ale yeast. In fact, for half of the chromosomes in lager yeast, or about half, it matches exactly, right? It is nearly exactly the same genetic information. For the other, and those are coded in blue here. The chromosomes coded in orange don't match that ale chromosome exactly. They're close, but they're different enough that they seem to be a different species. For example, you could line up the chromosomes of Cerevisiae and paradoxus also pretty well, but there'd be some differences. There's more differences between this unknown kind of chromosome and the ale yeast chromosome. And so people looked at this and said there are two different kinds of chromosomes in the cell of lager yeast. These probably came from two different parents. This is more evidence that our lager yeast is a hybrid. And so people thought, well, we don't know what the other parent is. We don't know what that yeast is. We have the DNA sequences of Saccharomyces paradoxus, of Saccharomyces cerevisiae, a couple of other ones. And we're always going out in the wild and trying to find wild Saccharomyces. And here's a global map that shows, in 2014, when I made this map, where people had found them. One thing I'd like you to notice is there's a lot of empty places on this map. These empty places are likely not to be places where there's no Saccharomyces. They're places people haven't looked yet. And so, the entire continent of Africa, people just haven't looked. There almost certainly is wild Saccharomyces there. India, people aren't really looking. I bet if they did, they'd find some. But Europe, people love looking for Saccharomyces in Europe, and China and Japan and the United States and Southern South America. And when they look, they generally find them. And they find many different species. So in 2011, some people went to Argentina and they sampled these southern beech forests. And they really like to sample this thing. This is actually a fungus. It's a big, visible to the naked eye fungus, but it happens to produce a lot of sugar. And they thought, well, if we're looking for Saccharomyces, maybe, a yeast that loves sugar, maybe we'll find it here. And they did. They found a Saccharomyces. They said, oh, cool, we found a Saccharomyces. Maybe it's Saccharomyces cerevisiae. But they sequenced the genome of that Saccharomyces. And they found that, in fact, it lined up pretty much perfectly with the unknown mystery chromosomes from lager yeast. And so they named this yeast Saccharomyces eubayanus. And they said, we've identified the parent, the mystery parent of our hybrid yeast. So this was the big discovery of 2011. Like, oh, we finally, after centuries, after centuries of having lager yeast, we now know where it comes from. It comes from Argentina. How did it get to Europe? And the answer to that appears to be, well, once we knew it existed, people looked for it elsewhere, and they found it in China. And the hypothesis we're working with now is that it was contact in the Middle Ages between people in China, in India, in the Middle East, all the way to Europe. There were trade routes, there was contact that allowed these two yeasts to come together, perhaps in a brewery, and make lager yeast. And here those yeasts are on the cladogram. They're pretty distantly related, and it's a bit miraculous that they can mate and make a hybrid at all. But this also helps us to understand they're so distantly related, why they can't really make gametes that survive long enough to have sex. All right, so that's our story about beer. And it's a little bit more complicated than our story about wine. Yeast comes from nature, right? And all yeasts come from nature, even the ones that we don't use for brewing. We learned that Saccharomyces cerevisiae is a yeast that you find both in wine and beer. That Saccharomyces pastorianus is a yeast you pretty much only find in beer, and that it comes both from Saccharomyces cerevisiae and from a wild yeast that you never find in domesticated environments, that they somehow were brought together and made a hybrid that has, to this day led to an enormous global economic impact. And of course, these aren't the only stories I can tell about fermented beverages. Fermented beverages are a worldwide phenomenon. Essentially, places people want to get drunk, they figured out how to ferment plant material to make alcohol. And I'd be really interested in learning more about the yeasts that are responsible for these fermentations globally. And my guess is they have equally interesting stories. Where did they come from? How did they get there? How do they interact with people, as beer and wine do? And so I'm really excited to learn more of these stories going forward. And I hope you are too. So we asked some questions about yeast. We asked how diverse they are. They are very diverse. Their diversity is similar to animal diversity. They are very widespread. You find them everywhere. We found them in forests. We found them on grapes. We found them in carnivorous plants. And other people have found them other places. They got into our food and drink from nature somehow, right? From grapes, from forests. So there's a lot of exciting work understanding how this diversity influences human beings. And with that in mind, I'd really like to thank you very much for your attention and for this invitation. It was a real pleasure to be here telling you about yeast.
Wonderful. Thank you, Primrose. That was exciting. We have a few questions from the audience, and then I have some of my own while the audience asks more. So one question from the audience is why does beer or wine production not use other sources of sugar like other cereals or other fruits, or are those also used? But is there some reason these specific ones seem to be preferred for mass production?
Yeah, I mean, the answer is other ones are used. Cider is an alcoholic beverage made from apples. These are used, I think, for a lot of reasons. They're traditionally used. People have a taste for that particular beverage. So people just like beer or like wine. Grapes have a lot of sugar in them, especially wine grapes. They're really sweet. The grapes that are used for wine are much, much sweeter than the grapes that you buy to eat as a dessert. So the plant puts a lot of sugar into these fruits. Cereals, it's the same thing. I mean, we grow cereals and have been growing cereals for food for a very long time. And we use the same cereals for beverages as we use for food. And, in both cases, we're using them because they have a high sugar content. So we're finding plant products that have a lot of sugar, and we're using them to make alcohol. This global map I showed you, these different alcoholic beverages come from different sources too, different plant sources.
So it's just a matter of some historical contingency then that these were not used for industrial production, because I mean clearly if you're going to rely on palm sap, it's a bit hard to make a mass production.
And it's used in a lot of different places, right? People have figured out how to make palm sap into alcohol. My guess is it's more what's marketable, what people have industrialized. I'm guessing you could easily, if people could in fact industrialize palm wine production. But palms take a long time to grow, and that there's economic questions, but
Mhm.
you know, I would love
Yeah.
to see more palm wine in my local supermarket.
Uh-huh. Uh-huh. So how different are the yeasts? So you said that both the ale and the wine yeast are Saccharomyces cerevisiae. Are they very different given that they've both been domesticated for so long, differently? And are they also different across different geographical locations where people have made wine, for example?
Absolutely, yeah, they are different. The beer yeast all seem to be very closely related to each other. And the wine yeast are a little bit more distantly related to one another. And this kind of makes sense if you remember that the wine yeast comes from nature. Um,
Mhm.
People have done experiments where they've made wine using beer yeast and
Mhm.
using sake yeast and using palm wine yeast. And what they found is that if you make wine with yeast that's kind of generally used for a different kind of beverage, the wine tastes bad. Like
Mm.
it's drinkable, you know, it has alcohol in it. A person could get drunk off of it. It's safe to drink, but it tastes bad. And so there's some thought that people have actually been selecting for, you know, what tastes good in beer and what tastes good in wine are not the same flavors. So people have
Mhm.
been selecting for those flavors.
That makes sense. We have a technical question from a participant who's watching on YouTube. Why can't yeasts mate with non-siblings? Or are those particular yeasts that you spoke about unable to do
Yeah,
so?
they can. And if you bring them together in the lab, they do. It seems that they prefer to mate with siblings, or
Mhm.
maybe it's that they can't find an unrelated mate. So yeast can't swim, they can't really move around themselves. And so one thought, and I'm really thinking of Saccharomyces paradoxus here. You see
Mhm.
much more mating with non-siblings in Saccharomyces cerevisiae. And the
Mhm.
explanation we have for that is that, well, people mix up Saccharomyces cerevisiae, right? If you're making wine, you've got your grapes from this side of the field and that side of the field all mixed up together. So
Mhm.
maybe we're bringing them together, but in the forest, they don't have that opportunity. The truth is we don't know. That's
I
our
see.
best guess. Yeah.
Uh-huh.
Yeah.
Uh-huh. And could you say something about what would happen in these experiments with wine, where you said that people replaced with some other yeast, right, that has also been domesticated, it tastes bad. But if you allow the process to sort of be such that the regular wine winner, the Saccharomyces cerevisiae, is not able to outcompete the others, and let's say one of the others who initially starts off, you let it sort of take over, does that also change the wine quite substantially?
It does. It would. So this is actually very difficult to do because
Mm.
you know, the Saccharomyces cerevisiae, it really does take over every time. So what you'd have to do is you
Mhm.
have to sterilize it and then add those other yeasts in. And people
Okay.
have done this sort of thing. They make you get alcohol, you don't get as much alcohol. The flavor is different. A lot of winemakers, they really want to encourage the growth of these early yeasts because they
Mhm.
add different flavor. Like each yeast makes its own flavor molecules.
Mhm.
And winemakers often want those flavor molecules or they want to exclude them. So winemakers are carefully planning, you know, maybe I can keep the temperature down long enough so that those other yeasts can survive longer, so that I can
Mhm.
get these other flavor molecules. And
Mhm.
some of
Mhm.
these flavors, you know, because I've worked with these yeasts, I've smelled them, you know, just growing them in the lab, they have really
Mhm.
strong smells. And
Mm.
sometimes the smell is delicious and sometimes it's not good at all. And so the winemakers, you know, they're being very careful. They're like, well, how hot do I want it to get? If it gets too hot, I know everything else will die besides Saccharomyces cerevisiae.
Right.
You know, how long, you know, can I keep it cool? Can I refrigerate it? You know, you know, do I want a big vat or a little vat? You know, they're making these decisions. And I think this is, you know, wine is often, it can be a cheap or an expensive product. If it's expensive, it's often expensive because a lot of work went into it. You froze a bit. I hope
There's also a question about, oh, can you hear me
I can.
now?
Yeah, I just wanted to make sure we were still connected.
Oh, okay. Yes. Maybe I'll shut my video off too, in case my internet is bad. So there's another question, and then maybe I'm paraphrasing here from Sowdhamini. Could you say something about wine tasting? So, for example, when people, who are connoisseurs of wine, say that they can tell different wines apart, is it known whether those are flavors directly imparted by the yeast products that you were just talking about, or is it also coming from the substrate that is used as the sugar? Or other aspects of the process of fermentation. Is that something we know about?
Yeah, it is. And I'm going to get myself in trouble because I'm not a wine taster. But what you're tasting when you taste wine is something called secondary compounds. These are molecules that living things make. So yeast, Saccharomyces cerevisiae makes secondary compounds that have a flavor and a smell. Grape plants make secondary compounds that have a flavor and a smell. Yeast sometimes will take the grape compounds and change them a bit so they have a different smell. I didn't talk about it, but there are bacteria in wine, and they also make secondary compounds. Again, these molecules that have a taste and a smell. And so it's a complicated question. It's coming from everywhere. The grapes, the yeast, the bacteria, the different species of yeast, the different species of bacteria, the interaction among them, because one microbe can change the molecule that another microbe makes. So there's just like all sorts of things going on. And I think people train themselves. I'm not well trained. So you could give me a delicious glass of red wine and I'll say, oh, that's a delicious glass of red wine. That's great. The people train themselves. They practice smelling certain molecules so they can recognize them. And sometimes they know where the molecule comes from, exactly what it comes from. And sometimes, it's a bit mysterious still.
Yeah, fair enough. So there's another question following up on the earlier question about inbreeding in these yeasts. Do we have evidence for inbreeding affecting the fitness of these, sort of obligately asexual guys who've been making lager for us for a long time? Is there any hint of that?
I mean, this yeast is very, very successful. And it does not mix up its genetic material at all, Saccharomyces pastorianus. Maybe if it could mix up its genetic material, that mixed up pastorianus would be even more successful. And with enough growth and enough, living in the world, it's quite possible that ability will return. It can make gametes, they just don't survive. One thing that's really remarkable about Saccharomyces pastorianus is there are two kinds, and that's it. So it seems like there were two hybridization events, and that's it. That's all the genetic diversity. There have been some mutations, so some random changes in the genome over the past few centuries. Sometimes if you look at the figure, you see that the chromosomes have sometimes they've swapped bits. You can see that right here, they've swapped bits. But for the most part, there have been very few changes to these chromosomes in the past few hundred years. I mean, fitness is a relative thing, and it's fitter than anything else in that habitat. It grows better at low temperatures than Saccharomyces cerevisiae does. Human beings really like it. And because human beings really like it, it's fit because we are moving it around. I don't know if it would.
Fair enough. Yeah. But I guess the question is could it, right? Maybe we have to put in more inoculum because of these problems with inbreeding that we do see, for instance, or we might be able to make beer even faster if they were able to grow faster and do the process of fermentation better if they weren't inbred. So I guess that's kind of,
no, maybe. So people have tried to they've tried to improve yeast. Brewers are really interested in making better yeast. And the problem they have, actually, this is a serious problem, is you can do that with cerevisiae and you can't do it with paradoxus unless you use, modern genetic engineering techniques. Because with cerevisiae, you can say, like you would with cattle, right? Like you would with some kind of domesticated animal, you breed them together to get some trait you want. And you can do that with ale yeast, no problem. You simply can't do do it with lager yeast.
Right. There's a question going back to the ecology of these some of these yeasts from Venkat, who's here on Zoom. So he's asking how, I mean, do we have evidence for climate change affecting wild yeasts, and could you say something about their impact on food chains? You know, who eats these wild yeasts, and could it have major impacts?
So these are fantastic questions, and he's actually touched on some of my research questions. So we don't know, is one answer. I can give you some hints of what we know. So the food chain one, it appears that the thing that eats these yeasts is insects. You know, we don't know how specific the insects are, or even which insects. It seems like wasps eat them. Maybe fruit flies. It's hard to say. We don't know anything beyond that. In terms of climate change, I think this is a really interesting question. So people have found out that the thing that determines if you go out into a forest and because they've done these studies in Europe and North America, they're really talking about European and North American forests. If you go out into a forest in Europe or North America, you will either find wild Cerevisiae or wild paradoxus, depending on the average high temperature in the summer. Cerevisiae likes it warmer, paradoxus likes it cooler. So you might expect that with climate change, Cerevisiae is going to start moving north. And one of my research questions that I want to research and don't know the answer to is, what happens when Cerevisiae moves north? Is that an opportunity to hybridize more? Is that an opportunity, or does paradoxus die off? Or how are they competing, or are they competing? So I think these are really interesting questions, and this is absolutely relevant to climate change. And I don't know the answer because nobody knows the answer yet.
Yeah. And does beer making in tropical places have extra challenges apart from, well, I guess obviously the fermentation has to happen at a given temperature, but for the yeast, but have people adapted different beer yeasts to the local climates?
Ooh, that's a good question too. I mean, one thing that we have is refrigeration. And so if you can afford refrigeration, then you can do the exact same thing no matter where you are in the world. I think, you know, if refrigeration is an economic challenge, then I think you are going to be challenged to think about, well, which yeast should I use so I, you know, minimize the amount of refrigeration I have to use. And one thing I'd love is to see, you know, brewers in tropical countries adapt the yeast from local beverages, so things like palm wine. You know, and to it's very trendy nowadays in the United States for people to go out into the forest and find some random species of yeast and try to make beer out of it, like completely unrelated to Saccharomyces. And sometimes they're successful, and sometimes, like, mostly it tastes bad. You know, like, I wonder, you know, how somebody who lives in a place where there is a local beverage that's not beer could adapt that local beverage to industrial brewing. And I'd love to see the outcome of that.
Yeah. Well, I saw something along those lines, not quite industrial, but you know, bigger than what you could do in a single house, usually at a household level, in northeast parts of India, where they make sort of beer, sort of wine from rice very often, where they grow very different kinds of rice varieties as well. And so on a project looking at rice microbiomes we had gone to collect, there was this lady who was sort of had these big tubs of rice beer / wine, you know, brewing, and I think she sold it for medicinal purposes as well, like a local traditional medicine of sorts. So I guess alcohol content was very low. It was tasty. I tried it, and they would have these inocula made of I think a plant root which they harvested locally. Plus, you know, and so you get these like cakey thing at the end of, I guess, one round of fermentation, and they would use that. So it seems like they have adapted it really nicely to at least some scaling up of some sort to be able to sell it. So yeah, I guess there's a lot to be discovered in many of these traditional brewing places, so people do it.
Yeah, I think so. And I'd really love to see some of those beverages scaled up. because I think that, you know, I think beer and wine are great. I think globally they get a lot of attention that other beverages deserve.
Yeah. Okay. I guess we are perfectly on time at this moment. I'm sorry, there are a few more questions that we couldn't take right now. But thank you very much for asking all these questions, and thanks to the participants. And I think I'll hand over to Sowdhamini to do the final set of thank yous.
Yeah. I'm not, yeah, so thanks for a very interesting talk, Primrose. And I'm sure we all have a different viewpoint about beer and wine after hearing your talk. It's very, very interesting and informative, and got us thinking about how yeast is all around us. Thank you so much. And thanks, Deepa, for co-hosting this session, this webinar. And thanks to the communications team for supporting this series as ever. And thanks to you, audience, for being so curious and asking some fantastic questions as ever. And yeah, with this again, I would like to thank the speaker and for giving an interesting talk today. And to the audience, yeah, we will meet next week on Sunday, but at 11 a.m., right, Pavitra?
No, we are meeting at 4 p.m.
4 p.m. Yes, yeah, right. Yeah, because we have speakers from all around. That's great. Until then, please take care. See you.
Okay. Thank you very much. See you. Bye, Primrose.
Bye.
It's a pleasure. Thank you so much.
Thanks. Bye.
Bye.